Hello, dear students. Today we will talk about special chromosome, which is a subtopic of chromosome chapters under plant cell biology unit, which is included in our botany semester 5th final year students of BSc. Before going to actual topic, let us once recapitulate the position, shape, and size and morphology of a normal chromosome in eukaryotic organisms. In eukaryotic organism, DNA molecule acts as a main genetic material. This DNA molecule having two long polynucleotide chain with a diameter of 20 angstrom. This DNA molecule wrapped around the histone proteins. It is the first level of organization of chromatin. This nucleosome consisting 10 nanometer size. This nucleosome fiber undergo coiling. It makes 30 nanometer size solenoid structure. This solenoid structure consisting 6 to 7 nucleosomes. These solenoid structure undergo supercoiling process. It makes 300 nanometer chromatin fiber. This chromatin fiber highly condensed undergo supercoiling and highly folded. It makes a metaphase chromosome. So in this fashion, we can observe the DNA molecule is highly condensed and organized in the metaphase chromosome during the cell division process. In the normal state, in the normal state, in the normal state, in the normal state, within the nucleus, the chromosomes are not seen clearly or distinctly because all chromosomes are appear as thread-like structures which are coiled with each other, and it make as a chromatin material, not chromosomes. So what is a special chromosome? The entire eukaryotic organism consisting of a normal chromosomal set carrying the genetic material in each cell. Each chromosome carries some genes from parent to offspring. This chromosomal set is specific to a particular species, but it varies across the kingdom. In eukaryotes, chromosomes are to standard structure and function of chromosomes as observed in mitotic and or meiotic cells in plants and animals. And during the cell division process, the chromosomes having some special features and the size, shape, and structure of the chromosome seen clearly in the metaphase or anaphase stage. But in some eukaryotes, besides the possessing the usual type of chromosomes, contain some unusual chromosomes. These unusual chromosomes are present only in certain special tissues, not seen in other tissues, and at some particular stage of their life cycle. Normal chromosomes are seen almost all in all cells, but in few certain special tissues, which are not seen in all species, in few particular species or animals at the particular stage of their life cycles. In these organisms, there are special tissues in which chromosomes differ in size, shape, structural specializations when compared to normal one. So the special type of eukaryotic chromosomes are classified into following types. One is Lamb-Brush chromosomes, another one is Polytene chromosomes, third one is B chromosomes, third one is B chromosomes. First of all, we go to the Lamb-Brush chromosomes and Polytene chromosomes. Generally, these two chromosomes are called as joint chromosomes. These chromosomes are decondensed during the interphase stage, but some exception and highly specialized chromosomes of vertebrates and Polytene chromosomes of insects. In both these chromosomes, the region that are actively synthesizing RNA are least condensed. In the least condensed stage, the DNA molecule actively synthesizing mRNA transcripts. And joint chromosomes are very long and thick during the metaphase. Hence, they are known as joint chromosomes. First, Lamb-Brush chromosomes. First time Lamb-Brush chromosomes are discovered by Ruckert in 1892. These chromosomes occur in oocytes of vertebrates as well as in some invertebrates also. These chromosomes found in oocytes which produce a lot of RNA and their cytoplasmic and nuclear volume increases. The nucleus size and cytoplasmic and nuclear volume increases comparatively. The detailed structure have been studied during the diplotene stage of meiotic division. During the meiosis process, the prolonged prophase having diplotene stage. In this diplotene stage, certain chromosomes stretch out large loops of DNA, causing the chromosomes to resemble a Lamb-Brush structure. In the diplotene stage, these chromosomes are stretched out with large loops of DNA. It looks like a Lamb-Brush model. So that's why these chromosomes are called as Lamb-Brush chromosomes. Moreover, they are observed under the microscope, light microscope, because the size is enormously increased. So that's why under light microscope also we can recognize. See here, this is the Lamb-Brush chromosomes. So this is the chromosomal chromatid. On the axis of chromatid, loops are there, lateral loops are there. These are the lateral loops. And the last tip of this chromatid, it is nothing but is called the telomeric region, or a Lamb-Brush chromosome with the telomeric loop. And these are the lateral loops or side loops. And without any loops, that is called as chromatid axis. The morphology of Lamb-Brush chromosomes is it consisting of a main axis having two chromatids. During the meiosis division, the two homologous chromosomes are pairing taking place. That is known as synapsis. So it consisting a main axis having two chromatids. Main axis has a row of granules known as chromomeres, which are held together by fine axial fibers. So along the chromosomal axis, the fine granular structures are there. These fine granular structures are called as chromomeres. So these chromomeres develops lateral loops in pairs. About 1 to 9 loops may arise from a single chromomere, but the size of the loop is vary from place to place. They are held together at points of chiasma formation. Chiasma means where a point, the two homologous chromosome attached, that place or position of the chromosome is known as chiasma. Chiasma means is nothing but the two homologous chromosomes are attached at a particular point. The loops of a paired chromosome form mirror image structures. And just you can observe the two lateral loops are exactly mirror image structures. This stage can last several months also. So just you can see that electron microscopic photograph. This is a chromatic. This is another chromatic. Okay. Both the chromatids having the granular structures. Each granule that is known as chromomere. These chromomeres are produced to lateral loop structures. This is the central axis. These are the lateral loops. So by the diagrammatic way, we can see this one. So that is the axis, both the chromosomes, where these two chromosomes attached at a point that is known as chiasma. So in the chiasmatic region, the lateral loops are developed. These lateral loops, which are symmetrical structure, both the lateral loops are same as like mirror images. So this is the axis and it is 10 micrometers. These extended loop-like structure containing highly condensed with heterochromatic region. The transcription occur either along the whole loop or at parts of loop. At the beginning of meiosis, when DNA replication is complete, the homologous pairs lie immediately next to each other, form characteristic structure composed of 4 chromatids. Lamb-brush chromosomes are distinguished by an especially high rate of transcription. What are the functions of Lamb-brush chromosomes? Lamb-brush chromosomes are involved in the synthesis of RNA and protein molecules. Each loop is believed to represent one long operon consisting of repetitive cisterns. Each locus codes for RNA. The loop is supposed to synthesis at high rate because of repetitive gene sequences. That means the nitrogen bases, either GC or AT rich sequences are present in this locus. There are also reports that the Lamb-brush chromosomes helps in the formation of yolk material in the egg. So whatever the protein material and enzymes synthesized by the RNA, which helps in the development of embryo. So second type of chromosomes is known as Polytene chromosomes. These Polytene chromosomes are discovered by E.G. Botany in 1881 in squash of salivary cells of Chironomus, as well as in Drosophila also it is investigated. They also occur in rectal epithelium and Malpighian tubules. So these Polytene chromosomes are also called as salivary gland chromosomes. So most of the insects belongs to Diptera. They possessing this type of abnormal, unusual chromosomes. They are many times larger than the normal chromosomes, reaching a length of 200 micrometers and are visible even under a compound microscope also. These Polytene chromosomes are formed during early larval development when meiotic divisions come to end in these tissues. According to an estimate, the polytene chromosomes have 1,000 times more DNA than the normal somatic chromosome. For example, a chromosome containing two DNA molecules which are organized in two chromatids. But during the meiosis process, the DNA undergo several cycles of replication. That's why it produced a number of DNA copies. Number of DNA copies. These are all DNA copies are situated in chromatid only. Because of these chromosomes actually consist of many strands, many chromonemata strands. That's why it is called as polytene chromosomes. Polytene chromosome. Poly means many, chromonemata strands are present in the chromosome. So, which containing 5 long and 1 short arm radiating from a central point called chromocenter, formed by the fission of centromeres, all the 8 chromosomes found in the cell. So, in the Drosophila, 8 chromosomes are there. These 8 chromosomes formed a chromocenter by the fission process. Of the 6th arms, the short arm represents the fused 4th chromosome and the longest represents the fused 6th chromosome. About 80% of the DNA is located in bands and about 15% in interbands. A polytene chromosome represents a distinct pattern of transverse banding. It consisting of dark colored bands alternating with light color interbands. So in the band region, active DNA is present, and in the interbands, inactive DNA or inactive genes are present. Intensively stained chromosomal segments correspond to high degree of packing or genetically inactive, that is heterochromatic. Less tightly packed segments stain less distinctly and corresponding to segment with genetic activity, that is euchromatin. In the Drosophila, 5,000 bands have been found in 4 chromosomes of salivary gland cells. Chromomeres in bands are at right angle to the long axis of chromosome. So these bands have a high DNA content and absorb UV light also, ultraviolet radiation. Painter and Bridges showed that in the Drosophila, the bands are associated with genes. So just found in the salivary glands and other tissues of flies, which is seen under the microscope in the nucleus during the interphase. So see here, these are the chromomere, the polytene chromosome, which have a number of many strands. The fission center is called as chromocenter. So this is called chromocenter. So 4 chromosomes are attached, fission taking place. So here light color band and thick color bands are present. The band and interbands are present on the chromosomes. So functions of the polytene chromosomes. Polytene chromosomes form structure that correlate with the functional state. During the larval development of Drosophila, a series of expansion appear in temporal stages in the polytene chromosomes. In certain places, the chromosome develop lateral loop-like structures. They are called as chromosomal puffs or Balbiani rings. These chromosomal puffs are decondensed, expanded segments that represent active chromosomal regions. So in this region, the DNA genetic information is transcribed. The location and the duration of the puffs reflect different stages of larval development. The incorporation of radioactively labeled RNA has been used to demonstrate that RNA synthesis, it is a sign of gene activity, that is transcription, occur in these regions. So this is called Balbiani rings or chromosomal puffs. There are certain interesting structure associated with the bands in the giant chromosomes called as chromosome puffs or Balbiani rings. The swellings are called as chromosomal puffs. The puffs are associated with the metabolic activities, represent area of active RNA synthesis takes place. So these are chromosomal puffs. At certain places we can see this swelling of the swelling part of the chromosome. So these are Balbiani rings. So in this region, RNA transcription takes place. Increasing the volume of the cell's nuclei, causing the cell expansion. A metabolic advantage as multiple copies of genes permits a high level of gene expression. In Drosophila melanogaster, the chromosomes undergo many rounds of endo-reduplication to produce large amounts of glue before pupation. So the larval stage converts into pupa. So in this metamorphosis, a glue product are necessary, which produced in large amount due to the endo-reduplication of the chromosome. Endo-reduplication means several times the DNA undergo replication process. It produces number of chromonemata strands. There is a tandem duplication of various polytene bands located near the centromere of the X chromosome, which results in the bar phenotype of kidney-shaped eye in the Drosophila. So next, third type of chromosomes, B chromosomes. All somatic and sexual chromosomes present in a diploid cell are known as A chromosomes or primary chromosomes. In addition to A chromosomes, more chromosomes present in a diploid cell, which are dissimilar to normal A chromosomes, is known as B chromosomes. Sometimes they are called as supernumerary chromosomes. These chromosomes lack homologous part. That means there is no homologous chromosome for the B chromosome. They identified mostly in plant species, highly in Gramineaceae members, and animals also, but the number of B chromosomes varies because the number is different from one species to another species. B chromosomes are majorly heterochromatic and non-coding region. These chromosomes are not essential for the plants, but these chromosomes containing inactive genes and which are non-coding regions. Due to the non-disjunction of chromosomes during cell division at the time of pollen formation. How B chromosomes are arise in the diploid cells? During the meiosis process, non-disjunction of chromosomes, that is unequal division of the chromosomes in the anaphase, a gamete receives n-1 chromosomes, another gamete receives n+1 chromosomes. In this fashion, a n+1 gamete fusion with n gamete, it leads to formation of 2x+1 product. In this 2x+1 product, along with A chromosomes, one extra chromosome is present, that is B chromosome. These chromosomes observed in all aerial parts, not in root system. See here, just this is the during the anaphase process, one of the chromosome is alone, which not move to the upper pole. It will be retained in its own place. So these are the isolated single a part of chromosomes are called as B chromosomes. The B chromosomes suppress the synapsis process, which reduce multiple pairing between the homologous chromosomes in allopolyploids. If the B chromosome number is more, which influence on fertility of pollen grain and also seed germination. Flowering process is delayed in B chromosome containing plants. B chromosomes are not essential for the life of a species. It is not involved in Mendel's inheritance also. There is no rule in all cells having B chromosomes in a plant species, but few cells are containing B chromosomes. Thank you.